All right, so today we are going to be, last week we started looking at sugars, carbohydrates, and we learned about monosaccharides, which would basically be the sugars which are just made up of one molecule, one sugar molecule alone. And then we also learned about disaccharides, which are when you add two sugar molecules together. So today we are going to be concentrating on polysaccharides, and we are talking about when we have more than two sugar units joined together, right? So polysaccharides have more than two monosaccharide units per molecule. So, well, the main monosaccharide that you learned about last week was glucose, all right? So we're gonna see that, yeah, glucose is the main monosaccharide for the polysaccharides that we will study, all right? So the polysaccharides that are studied for CAPE would be, you have to learn about starch, then you must learn about glycogen, and you must learn about cellulose. And all three are made of monomers of glucose. Starch and glycogen contain alpha glucose. However, cellulose contains beta glucose. So we will now study the structures of the polymers and see how their structures allow them to carry out their functions. All right, so guys, copy that. And I'm going to first let you know how the units are linked together. So for each monomer, each polysaccharide, sorry, you need to know how are the glucose units linked in it. I'm just going to give you a summary of the linkages. So here are the linkages. So we have times when our glucose units are linked from the number 1 to the number 4 carbon. That's called an alpha 1,4 linkage. We find that alpha 1,4 linkage in starch and glycogen. And whenever we want our molecules to have a branch, such as in amylopectin and glycogen, then we have to use another type of linkage called an alpha 1,6 linkage. And whenever we want to create tough fibers, we use beta 1,4 linkages. But all of these you're going to see as we progress through the class. All right, so make note of this, guys. And then we're going to spend time learning each polysaccharide one at a time. And seeing how the glucose units are linked together. Right? So let me give you some time to copy this. All right, so guys, make a note of the linkages. And then we're going to start off by learning about starch. So starch is the first of the three that we're going to learn about in detail. All right, so let us have a discussion about the first polysaccharide that we're going to study, which is starch. So starch actually is made up of two different compounds. So starch is actually a mixture. All right. Let me just give you an introduction as to what starch is, and we're going to learn about the two compounds found in starch. All right. So let's talk some more about starch. Starch is a mixture of two compounds. One compound is amylose and the other is called amylopectin. So these two compounds mixed together makes starch. Now we're going to learn first about the first component, which is in starch, which is amylose. All right. At the CXC level, we treated starch as just one substance, but now we know it better. All right. So the first component in starch is amylose, and amylose is made up of alpha glucose units, where you can see here that the number 1 carbon of the glucose is bonded to the number 4 carbon of the glucose molecule in front of it. So what we are seeing is a linkage between the carbon number 1 and carbon number 4. So remember, last week we would have gone through, so this class we did last week, where we actually went through the structure of glucose and the number in the carbons. So that class would have to look at this. But essentially what we're seeing here is that the glucose units are joined together by linkages between the number 1 carbon and the number 4 carbon of the one in front. Number 1 to number 4. Now, this might have hundreds of glucose molecules, but we're not going to draw all the hundreds of them. So what I have done is drawn 3 molecules, and that is enough to represent a long chain. So if you draw 3 molecules and you have the square brackets with the N outside, this suggests that this is just a segment of the chain that we represent. And it has 1 4-glycosidic bonds. So in the amylose compound, the glucose units are joined by alpha 1,4 linkages. The chains, however, we're going to talk about it, after they link up, it doesn't remain like a nice straight chain, right? Because the glucose molecules have a lot of OH groups. And these OH groups that you see here, these OH groups, are going to cause the chain to coil up, where they form hydrogen bonds with other glucose units, which are not their neighbors. And so this molecule, amylose, in nature is going to exist as a helix, all right? You'll soon see the helix and everything, but I'll allow you time to copy the structure so far. Yes, you have to draw the actual structure of it. And then we're going to see how this molecule actually coils up in nature to form the helix. Yeah, yes, actually, the helix is similar to the DNA shape. Yes. And I should say that amylose makes up 20% of starch. And as we're saying, we notice that the chain does not stay straight, that the molecule is actually going to coil up to form a helix, which I'm going to be showing you the helix in a moment. So I have a section of the helix here, which you're going to have to sketch. And it's basically showing that this is the coil. So these hexagons represent the glucose, and you can see it's turning. It goes and it turns against the helix. And what we have found is that typically after every 6 or 8 glucose units, that's when it actually turns. And these dotted lines that you see me drawing in the helix represent hydrogen bonds. So it's hydrogen bonds that are formed between the hydroxyl, the OH groups, of the glucose units that maintains the shape of the helix. Yes, go ahead with the question.
Miss, could you go over that again, please?
All right. So remember now that we were saying that the molecules are joined by 1,4 linkages, but that a lot of OH groups are still available. So we did use the OH group at number 1 and number 4 to make the link. But we have an OH group on carbon number 2, we have one on carbon number 3, we have one up here at carbon number 6. So we actually have a lot of OH groups available on each group of molecule. So what we have found is that the OH groups, like for example, this OH group here can bond with another OH group on a glucose, and that glucose is like 6 or 8 molecules away from it. And that's what the dotted lines represent, hydrogen bonds between the OH groups on the glucose. And if you have these hydrogen bonds forming for every 6 or 8 glucose units, or the glucose unit forms a bond with a glucose that's like 8 molecules away from it, that's what ends up causing it to form this coil, right? And what you're seeing in the coil, what I've drawn here, are some iodine complexes, and I'll explain to you why I've drawn the iodine complexes there. Go ahead, Akili.
So, Miss, why are the bonds between OH and H and O? Why are the bonds between two OH molecules instead of between an OH and an H?
Okay, so it is between let me just give a little bit there. So when it comes to hydrogen bonding, let me give an example. So if I have a glucose unit right here, let me not use the number 1 carbon. All right, so it has a OH. Now, the H that is attached to the O is slightly positive, and the O is slightly negative. So a glucose unit that is way down from it, like 6 to 8 molecules away, if it has a OH on it, then the H, which is attached to the O, is slightly positive. And this is because the oxygen is very electronegative. So what ends up happening is that, for example, the oxygen from this molecule, which is slightly negative, is going to be attracted to that hydrogen that's slightly positive. If the hydrogen does not have an oxygen beside it, the hydrogen is not going to be slightly positive. It's just going to be a regular hydrogen, and that regular hydrogen cannot make a hydrogen bond. It has to be a hydrogen that's attached to an atom like oxygen, and that oxygen is pulling away electrons from the hydrogen, so the hydrogen becomes slightly positive, and so there is no kind of a charge on it, and so this positive charge on the hydrogen is attracted to the negative charge on a different OH group from a different molecule. All right. So I see, just a moment, I see Elise, you've just joined. I know you explained that you have another class and it's just ending. So what I'm going to do, I'm going to come back here, but I'm just going to go back up so you can just take quick screenshots of what we've completed so far, so you can get perspective. So we're dealing with polysaccharides. Okay, so that's our first thing. And then we started speaking about starch. And we saw that starch has two molecules, amylose and amylopectin. And now we are looking more at the amylose, which it makes up 20% of the starch. We're looking at why it coils up into this helix. And I'm hoping it is clearer to you, Akili.
Yes, miss. Thank you.
So this is where we're at. All right, so I'm going to give you some time to actually copy the helix as best as you can. And we are seeing how these dotted lines, hydrogen bonds, is really between the OH groups of glucose units, which are like 6 to 8 units away from each other. And we're seeing that because of this coil that it makes, then actually there is space in the coil that when we are doing the starch test and we add iodine, it's actually in those spaces that the iodine molecules go. And when they fit into these spaces in the helix, that's what gives us the blue-black color that we use, that we get when we're testing this. All right, so guys, let me give you some time to copy this. Amylose. Akili saw your question and we're going to get to it shortly. So we're saying that in the starch test, I'm going to make a note. So in the starch test, we add iodine to a solution of potassium iodide. And this creates the triiodide complex. So that's usually what we say when we're doing the starch test. We normally say that we dissolve iodine in some potassium iodide. And that's what we normally add to the substance that we think has starch in it. So we're saying that when the triiodide complex with the three iodine molecule atoms go and fit in the coil of the amylose, that is when it gets a blue-black color. So really and truly, the starch test is actually testing for amylose. So the starch test actually tests for amylose, not the other compound that is found in starch. The starch has two compounds inside of it, amylose and amylopectin. When we do the starch test, it's really the amylose that we are testing for. All right, I'm gonna show you a video. And let's see if we can visualize it a little bit more. So let us see if we see, because we're talking about three-dimensional molecules, it's better for us to kind of see it.
Hi and welcome back to Free Science Lessons. By the end of this two-part video, you should be able to describe the structures of the polysaccharides which form starch. You should then be able to explain how the structure of starch relates to its function in cells. In a previous video, we looked at the structure of glucose. Remember that glucose is produced in plant cells using light energy trapped during photosynthesis. This means that glucose is a store of chemical energy, and the energy can be released during respiration. Now there is one key fact about glucose that you need to learn. Glucose is extremely soluble in water. That's because it contains a large number of hydroxyl groups. Hydroxyl groups are polar due to the small negative charge on the oxygen atom and the small positive charge on the hydrogen atom. This means that hydroxyl groups can form hydrogen bonds with water molecules, and this makes glucose extremely soluble in water. And remember that scientists call molecules like this hydrophilic. Now, because of its solubility, there is one big problem with glucose. If a cell contains a large amount of dissolved glucose, then this can cause water to move into the cell by osmosis. And you'll see why when we look at osmosis in later videos. So to solve this problem, plant cells store glucose as starch, and we're going to look at that in the next section. Okay, so as we've seen, plant cells store glucose as starch, and we find starch in starch grains. Now starch actually consists of two molecules. These are called amylose and amylopectin. In this video, we're going to look at the structure of amylose. And remember that in the exam you could be asked to describe this structure. The first idea you need to understand is that amylose is a polymer of alpha-glucose molecules. Now we've already seen that two alpha-glucose molecules can join together to form the disaccharide maltose. And remember that the glucose molecules are joined by a 1,4-glycosidic bond.
So guys, in order to put the glucose units together in the chain, remember that this carbon here, the first molecule, this is carbon number 1. And when the OH group is below the ring, this is when we call it alpha glucose, right? So both of them, the OH for carbon number 1 is below the ring. So both of them are alpha glucose. So as he's showing you here, to join these two glucose molecules together, we must remove a molecule of water from between them. So we take the OH from one and the H from the other. And that's how we end up forming the water. And then when we take away the OH and the H, all that's left behind is the oxygen. So now the oxygen is what now links these two glucose molecules together. So this is the alpha 1,4-glycosidic bond. So then he's going to tell us that this is what joins up the glucose. But after we have made all the alpha 1,4-glycosidic bonds, then we were saying in the notes that we still have a lot of these OH groups. Well, not number 1, but we're going to use up all the number 1. So there is OH groups, some of which they didn't show. But if we should draw out the entire molecule, we would realize that there's an OH group here, there's an OH group here, and there's even an OH group on the carbon number 6. So all these free OH groups that are present on the molecule still, that we never actually used up to create this bond, they now can form another type of bond. So this is a glycosidic bond, right? Which is a covalent bond, right? But we're saying that these OH groups can form another type of bond, which is what we call hydrogen bonding, where the oxygen of one molecule gets attracted to the hydrogen of a molecule that is some distance away. And it's not as strong as the glycosidic bond. But these bonds that are formed between the available OH groups now allow the molecule to twist up like a helix, right? And that helix has a space inside of the middle. And when we are testing for starch, if we create this reagent by mixing iodine and potassium iodide together, we get a molecule of iodine that can fit into the coils of the helix. And once that molecule fits into the coils, we get a nice blue-black color. And we can say, yes, this molecule, this substance has amylose because I'm getting that nice blue-black color. And the only way I could get it is if these molecules went inside the coils of the amylose. Right? Following? All right, so let's continue.
This is a condensation reaction because a molecule of water has formed. Now, if we join together a large number of alpha glucose molecules, then we make the polysaccharide amylose, and I'm showing you that here. Now, I'm just showing 4 molecules of alpha glucose. However, amylose can contain hundreds or even thousands of alpha glucose molecules. Again, you can see that the alpha glucose molecules are joined by 1,4-glycosidic bonds, and each bond forms in a condensation reaction, producing a molecule of water. The amylose molecule then twists into a compact helix, with hydrogen bonds forming between glucose molecules along the chain. Now, as we said before, starch is a store of glucose, so when the cell needs glucose, water is used to break the glycosidic bonds. And remember that this is called a hydrolysis.
Really showing us the helix. I think maybe in the next part, let's see if in part 2. All right, so what I'm going to do is let's see if we can see the helix of amylose. All right, so this is another video that she does a very good job of explaining topics as well for A-level. I'm going to kind of fast forward a bit. All right, so I'll just play it until it gets to the part where it deals with amylose. All right, so
welcome to another one of the A-level biology lessons for free with Ms. Estrick. Today we're going into the final lesson in the carbohydrate series, and that is polysaccharides. So the polysaccharides are created by condensation reactions, and you have multiple condensation reactions bonding together all of the glucose monomers. And the 3 polysaccharides you need to know are all created from glucose monomers. We have starch, which is found in plants and it's a store of glucose. Cellulose, found in plants and its function is for structural strength. Glycogen is found in animals and it's a store of glucose. Now that's surface level information, which you may have remembered from GCSE. We will be going through today much more detail about the structure and how that actually links to these three functions. So you'll need to have a copy of this table for your notes, ideally. It covers all of the key points that you would need to know from the specification, and it compares it across all three polysaccharides. So listen out for the monomers, the bonds, function, location, structure, and for the highest marks, it's linking that structure to explain the function. So you might need to pause now just to draw down that table. As we go through, slot in the information, and at the end we'll go through the answers. So starch, to begin with, is created from two polymers. Both of those polymers are created from alpha glucose though, and where the glycosidic bond forms is what creates a slightly different structure where you can get amylose and amylopectin. We've already said it's found in plant cells. Specifically, you find it in grains of starch inside of chloroplasts as an example. And its function is an insoluble store of glucose. So we'll look at amylose and amylopectin. Amylose is the first structure that we'll go through. So it's created by condensation reactions. In this case for amylose, the bond, the glycosidic bond, forms between carbon 1 and carbon 4. And this will happen over and over and over because it's a polymer, it repeatedly happens. Now that unbranched chain of amylose coils up to make a helix. So that is our structure amylose, but there's also amylopectin. Now this also is formed by
delve much into the helix. I don't want her to go on to start amylopectin just yet. All right, so let me come back to the note. Oh, and maybe I should just show you a picture of the starch test.
Testing for starches in food. We need a balanced diet to give us the energy we need to work and play. Starches serve as a quick source of energy. But how can you tell if food is a starch? We're going to answer this question in just a few minutes by performing a simple experiment. Before we get started, remember, never touch or taste anything in the lab and always wear your lab coat and safety goggles. Okay, let's get started. All right, for this experiment, we're going to need a small bottle of iodine and a paper towel. We're also going to need 10 small samples of different food. I'm going to place all the samples of the food on the paper towel. Then I'm going to take a drop of iodine and place it on each sample of the food and see if it changes colors. Iodine is a reddish-brown liquid that turns blue or black on a starch. Be careful with the iodine. It can stain your clothing. After all my observations, I'll record the findings in my data table. All right, here we go.
That the solution that he's adding is a mixture of iodine dissolved in potassium iodide. From what he's documenting, which with that some of the foods, when he added the iodine to it, it turned blue-black, right? So what it means is that these foods contain amylose. So the iodine that he added, potassium iodide mixed with the iodine, created this molecule. 3 iodine atoms joined together, and they were able to fit into the coils of the amylose. All right. Then let's proceed. So we're gonna see now the other molecule that we must speak about now, which is also present in starch, which is amylopectin now. All right, so let me give you the structure. So amylopectin is more complex than amylose because it has 2 types of linkages. Just a moment. All right. So this is the other compound that is found in starch. And in fact, the majority of starch is actually made up of this one. Amylopectin makes up to 80% of our starch molecule, right? It's a polymer of alpha glucose. So we know that the alpha glucose units are joined together by alpha 1,4 linkages. But then something else happens. After we have joined 24 glucose molecules together, then we create a branch. So basically what we mean by that. So let us pretend that this was glucose unit number 24. So we have 20 other glucose units that came before. Now, the number 6 carbon on the glucose then joins up with the number 1 carbon of a glucose, right? So because the bond right here is between a number 1 carbon and a number 6, it's therefore known as a 1,6, alpha 1,6. So we already have the alpha 1,4 that joins the glucose together, but then after every 24 glucose units, we do something else where we make the number 6 and the number 1 carbon join together, and that creates a branch. Now, when I show you another molecule of amylopectin, you'll see it more clearly, but for now, we're zooming in to see the linkages, right? So copy this for me, and then I'll show you some more about the amylopectin, this is the molecule that actually has branches. All right, so let me show you a bigger structure, but I never wanted you to copy this, but it gives you better perspective. All right, so guys, you can picture the amylopectin looking like this, right? Oh, sorry, I'm just seeing that there was, yeah, the amylose reacts with the potassium iodine complex. I'm just seeing that. All right, so guys, if you were to really look at a sketch of amylopectin, it would look like this. So you would see the straight chain, but then you see branches. So we're saying that the branches, the branch points are created whenever we have the number 6 carbon joining to the number 1 carbon of another chain. All right, so maybe you can go ahead and like sketch a piece of this molecule just to like to illustrate what's happening there. So just a sketch is fine. And so I'm going to be moving on after that to show you now. Oh, I need to describe it, and then we need to speak about how, why is a plant storing glucose by joining the molecules together to make these polymers? Just a second. So we have made the sketch of amylopectin, all right? So let me just give you a description, and then we're going to get into how it is, how these two molecules help the plant to store many molecules of glucose in a safe way. So amylopectin forms a branched molecule. It doesn't have coils like the like amylose. And thus doesn't form a blue-black complex. When tested with starch. So if you actually were to get pure amylopectin and you were to add the solution, the iodine dissolved in potassium iodide, you actually wouldn't get a blue-black color if there was only amylopectin there. But if both of them, if the mixture, both of them are there, then you will get that, the blue-black. All right. So we're going to get into the features of starch. So we've already studied the structure now. So we know we need to understand the features and how does the features or structure allows it to carry out its function. Every single time when you're asked a past paper question, they always want to relate structure to function. So let us get the structure part out of the way first. So what are the features? It is not water soluble, but when it dissolves, it forms a colloidal suspension. Starch is not sweet because it doesn't have many OH groups, because the OH groups are either used up in the glycosidic bond or they are used up in hydrogen bonds. It gives a negative test if you test it with Benedict solution. It cannot diffuse out of cells because it's large. It doesn't exert any osmotic action within the cell, so starch does not attract any water into the cell when it is stored. And the good thing about it is that the starch can be broken down easily to release energy whenever energy is needed. That glycosidic bond, the alpha-1,4, can be broken down fairly easily if we add water to the starch molecule, and we can get back the free glucose to be used. So this is the way that plants store glucose. They store it by joining them together to make starch, all right? So please go ahead and copy that for me. So we're basically saying that, guys, the starch molecule is really a way for the plant to store glucose, store many molecules of glucose without disrupting the osmotic balance in the cell. If it were to just try to store a lot of free glucose molecules, it would create a lot of problems. The glucose would come out of the cell because it would diffuse. It would attract water into the cell would swell up and burst. So the cell cannot store glucose in the regular form as glucose. It would be too dangerous, right? So what the cell has to do instead is that it has to form a polymer of glucose, which is not as soluble, it's not going to have any osmotic influence, and allows it to store many glucose molecules. But when it needs the glucose, it can break it down and get glucose. So you see that starch is actually well designed to meet the needs of the molecule, all right? To meet the needs of the cell, sorry. So after we have learned the structure, and now the next thing now, which Kate will really dig into now, is to see if you can link exactly how the structure is makes it adaptive to its function. So we've already kind of listed its function, but let us now see how its structure is adaptive to its function. So let us start off here. Glucose, the molecule of glucose contains a lot of chemical energy, which is stored in the carbon-carbon and the carbon-hydrogen bonds. Glucose is, when the glucose is free, it's very water soluble because it has a lot of OH groups, which are polar. And so they can form hydrogen bonds with water molecules, which I'm showing you here. However, because of the fact that it's so soluble, it's very osmotically active. And if it were stored in high concentrations in the cell, water would be pulled into the cell by osmosis. So to avoid this problem, polysaccharides are formed where many glucose molecules are joined to form a large insoluble and osmotically inactive compound. All right? So I'm going to give you a chance to copy that, and then I'm going to give you a short break. After the break, we're going to continue learning about the adaptations. All right, so go ahead and copy this, and then have a short break coming up. All right, guys, so now it's time for you to get the break. After the break, we're going to be continuing with seeing more about the structure, and we have to speak about the other two polymers. Remember, we have to speak about cellulose, and we also have glycogen. Starch is by far the more complex one. The others will be easy. All right, so let me give you a short break. After the break, we continue. Right, so the adaptations of starch now. So for it to do the function that we just stated a while ago, which is to allow the cell to store large amounts of glucose without affecting the osmotic balance, we say starch is very compact. It has hundreds of glucose molecules in a small space. It is also large, so it doesn't diffuse out of the cell, and it's very energy efficient. It is insoluble, and therefore it is osmotically inactive, and this allows a high concentration of glucose to be stored in the cell without pulling in water. It also can be easily hydrolyzed or easily broken down when you need the free glucose. And what we find is that it's much easier to break down the amylopectin than it is to break down the amylose, right? Because amylose only has, it's a straight molecule, right? So it only has two areas where enzymes can attach, which is either at the beginning or at the end. So amylose doesn't really have a lot of attachment points for enzyme, but luckily it only makes up 20% of starch. When we're finished with this, you're going to see amylopectin, because of the fact that it has branches, it gives way more, it has way more points of attachment for enzymes. So it's much easier to get free glucose from the amylopectin than it is to get it from the starch, all right? So I'm going to give you a chance to copy that. I have another video to show you on starch, and then we're going to be moving on to the next polymer, which is glycogen. So guys, as we're saying now, when we see the amylopectin, we're saying that amylopectin, because of the branches, has more points for the enzymes to be attached. So it's much quicker to get the glucose from amylopectin than it is to get it from starch, right? And that is a very important difference, right? Because there are times when animals need to be able to access that glucose very quickly. For example, in times of danger, for example, an animal trying to run from a predator, you want to be able to break down your starch quickly and get the glucose you need. So that is actually a very important feature. So let me just give you that note, and then we're going to move on to our next polysaccharide, which is glycogen. So there are only 2 points of attachment for enzymes for amylose, but for amylopectin with the branches, right, we can see that it's way easier because at each branch point, every time there is at the end of the molecule, an enzyme can attach. Let me just see if there's a question. Okay, Joshine Senior, just saw your comments. Is that enough? All right, so I'm going back up now. And then we're going to talk about glycogen. We're going to go back to that video we were watching. And we're going to learn about glycogen. All right, guys, are we finished? So let's look at glycogen. So now that we have spent so much time learning about starch, it makes it easier for us to now understand what we are going to do with the others, which is to basically learn their structure, then learn their function, and see how their structure allows them to be adaptive for their function. That's basically what we're going to be looking at. All right, so.
Hi and welcome back to Free Science Lessons. By the end of this video, you should be able to describe the structure of the polysaccharide glycogen. You should then be able to explain how the structure of glycogen relates to its function in cells. In the last couple of videos, we looked at the role of starch as a store of glucose in plant cells. We saw that starch consists of 2 molecules, amylose and amylopectin. Both of these molecules are polymers of alpha-glucose. Amylose is an unbranched polymer, whereas amylopectin contains a large number of branches. Now, just like plants, animals also contain a glucose storage molecule. In animals, the glucose storage molecule is called glycogen, and the major stores of glycogen are found in the liver and in muscle cells. I'm showing you the structure of glycogen here. glycogen is a polymer of alpha glucose. Most of the alpha glucose molecules are joined by 1,4-glycosidic bonds. However, glycogen also contains branches, and the glucose molecules at the branch points are joined by 1,6-glycosidic bonds. Now you'll notice that glycogen seems to have the same structure as amylopectin. The main difference is that glycogen is more branched than amylopectin, which makes glycogen a very compact molecule. And because glycogen's got a large number of branches, it's got a lot of free ends. This means that enzymes can convert glycogen back to glucose very rapidly. Now, this is important as animals often have a high rate of respiration, and the energy needs of animals can change rapidly. For example, an animal may have to move quickly to escape from a predator. If this happened, then the rate of respiration would increase, and glycogen in the animal's muscles could be rapidly converted to glucose to be used.
Right. So guys, remember, you're not expected to write any notes from the videos that I show, right? It's just for you to get a preview. Anything that I need you to write will be what I put on the whiteboard, okay?
Used in respiration. Now, we've already seen that glucose is highly soluble in water, and because of this, a high concentration of glucose in a cell would cause water to enter by osmosis. Unlike glucose, glycogen is insoluble in water, and this means that glycogen does not draw water into cells by osmosis. And being a large molecule, glycogen cannot diffuse out of a cell. So all of these features make glycogen an ideal glucose storage molecule in animal cells. Okay, so hopefully now you can describe the structure of glycogen and explain how the structure relates to its function in cells.
So that was just to give you a preview, right? So I didn't expect you to copy anything from this. All right, so we're going to talk about glycogen, and I don't even expect you to draw this diagram either. It's just basically there to show you that the glycogen molecule is actually similar to amylopectin in the sense that it has branches. So you have the glucose chains, and then you have branches, right? So remember, the straight chains would be alpha 1,4, and whenever there's a branch, it's alpha 1,6. However, though, the difference is that you're going to have way more branches in glycogen than what we saw in amylopectin. So basically, it's a similar structure. It's just that the glycogen is more branched than the amylopectin. And glycogen is found in animals, while amylopectin is found in plants, right? So like glycogen is like the animal version of amylopectin. So again, I don't expect to copy that diagram. I'm going to give you a simpler version of glycogen that you can use for your questions, right? So here we have yes. So here you can see that in the straight chains, we have the alpha 1,4 linkages. And whenever we have a branch, we have alpha 1,6, just like what we saw in amylopectin. So this is a representation of glycogen. I will have you copy. And we're just going to go straight into talking about how glycogen is adapted for its function. Once you're finished making that sketch. All right. I'm going to put up the adaptations. You can go ahead and start writing them, and then we'll discuss them after. All right, so let me give you some time to make note of the structure and the adaptations of the glycogen. So guys, as it concerns the adaptations, we see that glycogen is a very compact molecule. Because of all the branches, it can be packed very well. So it's very efficient in storing large numbers of glucose in our animal cells. Yeah. The next thing is that it is insoluble. So it's not going to interfere with osmosis in the cell. It's large, so it cannot come out of the cell. It is very branched, so it means that there are multiple points of attachment for enzymes that can hydrolyze the glycosidic bonds to release glucose. This is essential because animals have a higher rate of respiration than plants. The animals will more likely need to get the glucose faster than plants need it. For example, like we saw in the video where the zebra was trying to escape from a predator. So when animals need a high level of glucose, they need it very quickly. So they need to have a molecule that can allow enzymes to attack it very quickly and break it down and release the glucose units. So that's why animals don't store amylose. Amylose is the helix and it doesn't have a lot of attachment points. So because we know the needs of animals, that we need to be able to mobilize that glucose very quickly, the way that we store the glucose is in the form of glycogen, which is very branched. So it simply means that the glucose can be available very rapidly in means of that, right? So guys, we have one more polymer left. So let me just give you another 2 minutes to finish up with that. And we have to look at the polymer of cellulose, which is the last polymer here. So 2 more minutes. And then we need to learn about cellulose. Is everybody finished writing? Does anybody need more time? All right, so question.
Yes, miss.
Oh, you're saying yes, you need more time?
Yes, miss.
Okay. All right, so I saw a question in the chat. So is it that we're saying that glycogen is similar to amylopectin? Yes. They basically have the same linkages. Only thing I should have mentioned to you, so remember that we said that amylopectin has a branch after, do you remember after how many glucose does it have a branch? Look back at the notes and tell me after how many glucose. Yes, it was about after every 24 or something like that. So so for glycogen now, I'm going to tell you how often it has a branch. And that's about the only difference between them. So glycogen has a branch after every 8 to 12 glucose units. So let's make a note of that. That's a very, very important point. So you can see that definitely glycogen is way more branched than amylopectin, which is why it's way more easier to release the glucose units because the branching comes way more often, right? All right, so that leaves us with our last polymer. But before we do that, I'm going to go back to the video we were watching. Yes. So I had started it and we had stopped it with Miss Estro. As I said, she's out. I find on YouTube that she actually is very good.
It was formed by condensation reaction.
So we had started watching it and we had stopped right here. So let's just finish up this video. And she will tell us about the the cellulose as well before we start. So let's just finish that.
And this lower part you can see is 1 to 4 glycosidic bonds. And we can tell that because you have this straight chain. At this location though, we now have part of it branching off. And this is created by a 1 to 6 glycosidic bond. So here we have carbon 1 of this glucose molecule and carbon 6. And due to that location, it causes a branch. So amylopectin is quite a branched polymer and amylose is a straight polymer which spirals up. The next one then is glycogen. This is also formed from alpha glucose. This time though it's found in animals inside of the muscle and liver cells. The function is also an insoluble store of starch, so we are going to see some very similar structures. So we can see that glycogen looks incredibly similar to amylopectin. It has 1 to 4 glycosidic bonds and it has 1 to 6 glycosidic bonds. One of the key differences though is glycogen has even more 1 to 6 glycosidic bonds compared to amylopectin. So it's a highly branched polymer. So this just gives an overview of how highly branched this molecule is. It can still be compacted though, so you can fit large amounts of glucose in a small space. Lastly then, cellulose. This is the most different in structure and therefore function. Cellulose, the monomer is beta glucose this time, so the other structural isomer. Its location is in the cell wall of plants and it provides structural strength, so it prevents the cell from bursting when it becomes turgid with water. So the structure then, the beta glucose molecules formed by condensation reactions to create this long straight chain. but they only contain 1 to 4 glycosidic bonds. So you only have straight chains. Now those long straight chains of beta glucose line up parallel to each other, and they are then held in place by many, many hydrogen bonds. And that's what these blue lines here are representing. Lots and lots of hydrogen bonds. So if we were to describe the structure that we've got so far, you'd get a mark for describing that there are long, straight chains. There would be a second mark for pointing out that these chains are held by many hydrogen bonds to form a structure which we call a fibril. So all of these long, straight chains held by many hydrogen bonds, that is a fibril. Now, the way that this provides structural strength is because of the number of hydrogen bonds. So I've pointed out here, an individual hydrogen bond is weak. Due to the large number, collectively, they provide the strength to the cell wall. So that's it. That is our 3 polysaccharides, and we'll go through the table so you can just check you have noted down all of the key information. So first of all, the monomers. We have alpha glucose in starch and glycogen, and cellulose is beta glucose. The bonds that form: starch forms 1 to 4 glycosidic bonds in amylose, but 1 to 4 and 1 to 6 in amylopectin. 1 to 4 glycosidic bonds only in cellulose, and glycogen forms 1 to 4 and 1 to 6 glycosidic bonds. But it'd be worth noting that glycogen has more 1 to 6 glycosidic bonds than amylopectin, because that's going to come into explaining the difference between the structures. Function. So starch and glycogen are stores of glucose, whereas cellulose is for structural strength in the cell wall. Location. Starch is found in plant cells, for example inside of chloroplasts. Cellulose is also found in plant cells inside of the cell wall. And glycogen is found in animals in the muscle and liver cells. So the structure. Starch is made up of 2 polymers of alpha glucose. Amylose, which is an unbranched helix, is unbranched because it only contains 1 to 4 glycosidic bonds. Amylopectin is branched, and that's because it also has the 1 to 6, and that causes the branching. Cellulose is a polymer which is formed from long, straight chains, and those chains are held in parallel by many, many hydrogen bonds to form a structure called a fibril. Glycogen is highly branched, and it's even more branched than amylopectin because it has more 1 to 6 glycosidic bonds. So the last part then is the explanation of how the structure provides the function. So the fact that it's a storage molecule, starch, the helix shape of amylose means it can be compacted down, as if you were squashing a spring. You're squashing it, you're compacting it, and that's a benefit. It means you can fit a lot of glucose into a small space. It has a double advantage though, because it also has amylopectin, which is a branch structure, and having lots of branches coming off provides multiple exposed ends of the molecule, and that provides a larger surface area for enzymes to attach, and therefore they can rapidly hydrolyze starch back into glucose to release the glucose to the plant if it needs it. The last one here is insoluble. Now that is the same for all 3. So all 3 polysaccharides are insoluble in water, and that is an advantage because if it doesn't dissolve in water, it will not affect the water potential, and therefore it won't affect osmosis. Now that's a benefit because it means you won't end up having excess water moving into the cells, potentially causing them to burst, or in plants, making them become turgid.
So guys, she just did a nice summary. So the only thing left for us to write about now is cellulose. So we are learning about this, the cellulose molecule, which we saw some of it in the video. Now cellulose, unlike glycogen and unlike starch, cellulose is made from beta glucose, right? So in beta glucose, the OH group is for carbon number 1 is pointing upwards, is pointing above the ring. So if we put 2 molecules of beta glucose together, where the OH on carbon number 1 is pointing upwards, but the carbon number 4 is pointing downwards, we have ourselves a problem. It is difficult to make the 1,4 glyphosate bond in cellulose because we are using beta glucose, right? And the OH on carbon number is pointing up. So the OH, if we want to make a bond between carbons number 1 and 4, it is quite difficult because the OH groups are kind of far from each other. Because they are literally, one is pointing up, which is the one for carbon number 1, and the one for carbon number 4 is pointing down. So cellulose has to do something unique, which none of the other molecules had to do. To overcome this problem, to get these OH groups to be aligned so we actually can remove water from them, the molecule of cellulose actually has to flip the second one. So the first one stays in its original position where the OH group is pointing up. The second molecule has to turn upside down. So you notice here the oxygen is pointing up. Now the oxygen is pointing down. So the entire molecule has flipped upside down. So what it means now is that this group that was pointing downwards on carbon number 4 is now pointing up. And now that we have both of the OH groups pointing upwards, it's now easy for us to remove the water molecule from between them and to join the two glucose units to create the glyphosate bond. So for the molecule of cellulose, it does something that none of the other polymers had to do, in that it has beta glucose, and every other, and I say alternate, or every alternate glucose molecule has to flip upside down. So it is able to make these 1,4 linkages, all right? So go ahead and copy that part, and I'm going to show you what it looks like after when we actually make the linkage. Copy the structure, guys, and then we proceed. All right, so I'm gonna scroll up a bit now so we can see what would be what would be the resulting structure, right? After we have flipped every other glucose molecule so that we could get them aligned to remove the OH group, right? So now we are able to create the glycosidic bonds. But the glycosidic bonds now are going to end up being, some of them are going to end up being above the plane and some are going to be below the plane. So this is what we're going to end up with. So for the ones that were above the plane, of course, the glycosidic bond will be above, and then the second one will be below. So basically, the glycosidic bonds are alternating. Some above, then below, above, then below, right? And this is going to have a great impact on the structure of the glucose, all right? So let me give you a chance to finish that. So now, guys, once we have looked at the structure, well, we have to learn some more about cellulose's structure, and we're going to see how cellulose is adapted for its function. And the function of cellulose is not to store glucose in the cell. That's not its job. Its job is to give structural support. So it's kind of different from the other two polymers. All right, so adaptations of cellulose for its function. It is a polymer of beta glucose with a structural role in our plants, in plant cell walls. It is a very strong molecule, and so it prevents the cells from bursting when cells become turgid or swollen with water. So cellulose has a structural role in our cells. Now, The other feature or adaptation is that it's made up of thousands of beta glucose molecules joined by 1,4 glycosidic bonds. Remember that every other glucose molecule flips upside down? And at the end of it all, we end up getting a straight chain that has absolutely no branches. And because these chains are straight, it allows many of the chains to run side by side or to run parallel to each other and then link up by hydrogen bonds. So I'm gonna show you a piece of the structure of cellulose to show you how many cellulose units or many cellulose chains running side by side can link up. So you can see in this diagram here that this is one cellulose chain here, it's straight. So another cellulose chain can come right beside it because this is also straight. There are no branches, right? So you can end up having multiple cellulose chains coming beside each other, and then they link up with each other by hydrogen bonds. So guys, you don't need to draw all of this. You can just draw a segment to represent it, like maybe 2 chains or something, or you don't even need to draw a lot of hydrogens, but kind of need to have representation because in CAPE, whenever you're describing things, whenever a diagram can help, you also should provide a diagram. So this is how cellulose runs. Chains are straight, they can run side by side, and they can link up with hydrogen bonding. And that's really why cellulose ends up forming such a strong structure that the plants use in their cell wall. And the formation of the hydrogen bonds allow for cross-linking to occur between the chain. We're almost there, guys, almost there. Remember, for this diagram, you don't have to draw everything. You just need to draw a representation of it. So this is a simpler version of showing how the chains of the cellulose are linked together. So literally the lines represent the chains and these are just showing the links between them. So because of these links, then cellulose ends up being able to form very strong fibers. All right, I'm just going to show you a diagram, not something you need to copy, as to how the cellulose fibers end up creating the cell wall. And then I have a video to show you. So I'm not going too fast. All right, so the other diagram you don't need to draw it, just to illustrate something. So I want to show you here how it is that how we get the cell wall, right? So here are the glucose molecules. You can literally see the glucose molecules joined together to make a chain. And we are saying that you can have multiple chains joined together, and that creates a microfibril. If you have many microfibrils, then you end up creating a fiber or a macrofibril. And then many macrofibrils come together to make the cell wall, right? So it's a fairly complex structure, right? So you're moving from just linking the glucose units together to create these chains, putting these chains together to make microfibrils. Microfibrils make fibers of those, also those macrofibrils, and then those them together to make the cell wall, right? It's a very intricate design. But at the end of the day, what I did on the last 2 points is what you need to copy. Oh wait, did I mention? Okay, I already mentioned that the microfibrils form strong fibers. I already spoke. So the other thing, the other 2 points to make note of is that the cell wall is freely permeable to water, but it does offer a rigid support to our plant cells. And very importantly, cellulose is not broken down to give glucose in plant cells, because plants do not have the enzyme that can break down these glycosidic bonds, these beta 1 glycosidic bonds. They don't have cellulase. So when we eat vegetables and we say we're getting fiber, what we really mean is that we're eating cellulose. And because we cannot break down the cellulose, it just passes through our digestive system. But we can break down starch. We have enzyme for that, right? But just remember, the point of cellulose is that it's not to store glucose to be used by the plant. It's actually to be structured for the plant. All right, so we're basically almost at the end. As I said, this diagram here, I don't expect you to copy it. It's the points I need to copy. I have another video to show you about the structure of cellulose. And then I'm going to speak to you about the past papers that we are now able to cover based on what we did today. All right, so is there anyone still writing? Or are we finished copying the notes? Is there anyone still writing? All right, so let's just look at the video, last final video about structure of cellulose. And then we look at the past papers.
Hi and welcome back to Free Science Lessons. By the end of this video, you should be able to describe the structure of the polysaccharide cellulose. You should then be able to explain how the structure of cellulose relates to its function. Now, the first key idea you need to understand is that cellulose is a major part of the cell wall found in plant cells. We're going to look at how cellulose carries out its role later, but we're going to start by looking at the structure of cellulose. So far, we've looked at the polysaccharides amylose, amylopectin and glycogen. These polysaccharides are all polymers of alpha glucose. Now, cellulose is different to all of these, and that's because cellulose is a polymer of beta glucose. I'm showing you a molecule of beta glucose here. The key thing to remember with beta glucose is that the hydroxyl on carbon 1 points above the plane of the ring. Now, this presents a problem. I'm showing you here two beta glucose molecules side by side. If I want to form a glycosidic bond between carbons 1 and 4, you can see that the hydroxyl groups point in different directions. So when a molecule of cellulose is formed, every second beta glucose molecule flips like this. And now we can form a glycosidic bond between carbons 1 and 4. Okay, I'm going to add a third beta glucose molecule now. In this case, you can see that we do not need to flip this beta glucose molecule to form a glycosidic bond between carbon 1 and carbon 4. So remember that to form cellulose, every second beta glucose molecule is flipped. You also need to remember that cellulose is an unbranched polysaccharide. Okay, in the next section, we look at how cellulose molecules are arranged in the plant cell wall, and how cellulose allows the plant cell wall to carry out its function. Okay, so as we've seen, cellulose is an unbranched polymer of beta glucose, and every second beta glucose molecule is flipped. Now, because cellulose forms a straight chain without any branches, this allows cellulose molecules to get close together, and I'm showing you that here. Hydrogen bonds can now form between neighbouring chains like this, and because a huge number of hydrogen bonds form, this makes cellulose extremely strong. When cellulose chains group together, scientists call this a microfibril. Microfibrils then group together to form larger structures called macrofibrils. And finally, macrofibrils group together to form a cellulose fibre, and these cellulose fibres form the plant cell wall. So the key feature of cellulose is its strength, and this strength allows the cellulose cell wall to carry out its functions. The cellulose cell wall is also permeable to molecules, and a good example is water. Under normal conditions, plant cells contain a great deal of water. As water moves in by osmosis, the plant cell contents push outwards against the cellulose cell wall. The strength of the cellulose cell wall means that it can resist the outwards pressure due to the cell contents, and this prevents the plant cell from bursting. When a plant cell is full of water like this, it becomes rigid. Scientists say that the plant cell is turgid, and these turgid plant cells help to give the plant its upright structure. Okay, so hopefully now you can describe the structure of cellulose and explain how.
Right. So guys, that's actually it in terms of the polysaccharides that we studied today. So now we need to discuss the past papers now, all right, on what we have covered. Remember that we do the past papers as soon as we learn the topic, all right? That is the best way to do it. It helps to reinforce the content, and it also gives you guides as to how you study for the exam. All right, so two things for today's classwork I need to point out. Number one, for some persons who are new, is once you get into your Canvas, the Zoom code is always found up here. And here we have the course materials. So we have a book of notes, additional reading. All right, this is the book. We have one book for paper two papers. Well, this is a multiple choice book. So it has from 2023 to 2011. We have a book of additional notes that you can read, right, on each topic. And we also have a book of essay type past papers, all right? So the multiple choice past paper, book is here, okay? Now, why am I showing you that? Because we have actually completed a module. We've actually completed 2 modules. So we completed water and its properties. We did that, right? And we just completed the module on carbohydrates. So what now I would have done, if you go back up to the multiple choice quizzes, you see that actually now you are in a position to do a quiz, a multiple choice quiz. All right, so this multiple choice quiz, let me just walk you through it. Just a second. So how the multiple choice quizzes work, and remember, we have to do multiple choice at the same time we're doing essay because multiple choice is graded and as the same weighting as essay papers, all right? So how it works is that I tell you the question and the year and the question, and most times you have to use 2 devices. One device displaying the multiple choice book, and this one with the question. So you would identify the question, read it, and then select the answer. And so we have, as you see in this quiz here, we have 14 questions based on the structure of water and carbohydrates. So remember that you look for the questions in the multiple choice book, which is in the course material section. And once it is done, you have a week. So sometimes it's by now until next Monday, you must go in and do it. However, the classwork, which is due by tomorrow now, would be these past papers. So based on what we covered today on polysaccharides. So we have this question from 2013, question 1B. It's a diagrammatic representation of the molecular structure of a short section of the glycogen molecule. Use a labeled arrow to highlight the location of each of the following. So you're going to tell where on this diagram can you find the alpha 1,4 linkage and where can you find the alpha 1,6, right? Then in the space below, they want you to sketch the general shape of a glycogen molecule. You do not need to give details of the glucose structure. So remember that simplified version that I gave you to draw, that's what you could put right here as a general structure of glycogen, all right? Next question says amylose, which is in starch, and cellulose are both polymers of glucose, but they differ in their structure and their function in plant cells. With reference to one structural property, briefly explain the functional differences of amylose and cellulose in plant. So I would refer to the functional property of one functional difference. I would say that the amylose is helical, right? So it is able to coil up and it allows, it can be packed up compactly. So it basically allows the plant, to store large concentrations of glucose without affecting the osmotic balance. On the other hand, cellulose stays as a straight chain. It doesn't coil up like amylose does. It stays straight, and because it stays as a straight chain, it is able to form cross-links with other chains, which also then allows it to form fibers, and that allows it to be able to form a strong structural support for the cell wall. So one molecule actually wants to be there for storage of glucose in the cell. The other molecule wants to give structural support to the cell. So you want to discuss that for 4 marks. The next question says, you have this carbohydrate. Here you're seeing 2 glucose units joined by an alpha 1 glycosidic bond. You need to draw a third glucose, and you're going to show how the third glucose molecule could be linked by an alpha 1,6 glycosidic bond. So they actually want you to search for which one is the alpha 1,4, and they want you to draw these 2 and draw a third molecule of glucose that was added by a 1,6 link, which is why we're going through and showing you how the links are made. You see how the past papers are asked. So then you're asked to distinguish between an alpha glycosidic bond and a beta glycosidic bond. The alpha glycosidic bond would be found in starch and glycogen. The beta linkage would be found in cellulose. Cellulose is the one that had to fit every other glucose unit. So they have beta linkages. And the beta glycosidic bond would be found below the plane of the ring. The alpha glycosidic bond would be found above the ring. Also, comment on the difference in function between the carbohydrate with the alpha linkage and carbohydrates with the beta. The ones with the alpha linkages, they tend to be used for storage of glucose in the cell, right? So they are mostly for glucose storage, but the ones with the beta linkage are more for structural support. So guys, it's really these two questions I have for you to complete by tomorrow to submit, right? Make sure the work is submitted by tomorrow, guys. Past papers are not optional, right? It's a part of your class. So remember that this has to be done by tomorrow, and the multiple choice quiz will be kept open for a week, so that sometime from now to Monday, you have to go in and complete. Do you have any questions before we go? Any questions? All right, guys. So enjoy the rest of your evening. Please make sure the work is submitted by tomorrow, guys. As I've been telling you, we are starting to notify your parents. Once we do not see a submission on time, we will contact your parent because we want the work to be submitted. All right. Thank you, guys, and have a wonderful evening.